So our project was on the gut microbiome of different animals. Microbiomes are the types and proportions of bacteria present in an environment. The gut microbiome is the collection of microorganisms composed of bacteria, bacteriophage, fungi, and viruses that are present inside the gut of an animal. The gut microbiomes have been studied and found to have an effect on multiple physiological conditions. One of these conditions is irritable bowel syndrome, also known as IBS. In IBS, the microbiota Clostridium is in less abundance in the bowel. Also, IBS patients exhibit an increased firmicutes to bacteroidetes ratio. This can be caused by dietary changes, probiotics, and/or antibiotics. Therefore, this relationship is correlative rather than causal. Another condition is obesity and type 2 diabetes. In studies, it has been found that obesity and type 2 diabetes can be caused by increasing the number of firmicutes to the detriment of bacteroidetes. Having a higher proportion of firmicutes and 50% fewer bacteroidetes are two microbes that can cause obesity. From this information, it can be concluded that this relationship is correlative. Our question for the experiment was, how do the gut microbiomes of different species of animals compare to one another? We compared the microbiome of pigeons and iguanas. There are very limited similarities between the two. The pigeon is a bird, and its diet consists of grains, seeds, berries, and insects, while the iguana is a reptile, and its diet consists of fruits, flowers, and leaves of plants. Through specific methods, we determined the nucleotide sequence of both animals by isolating the DNA of their microbiome. First, we isolated the DNA from the fecal sample using the QIAGEN kit. Then, we increased the concentration of the DNA by reducing the sample volume using the Zymo kit. We determined the concentration and purity of our DNA samples using the UV spectrophotometry nanodrop. The A260 to 280 values tell us the purity. Next, we used gel electrophoresis to separate charged macromolecules based on their mass and size. We combined each amplicon PCR reaction with the sample buffer. Then we loaded the samples into the wells of the gel. We started the first PCR cleanup by bringing the AM Pure XL beads to room temperature and mixed them with the sample to ensure binding of the DNA to the beads. We placed the PCR tubes on the magnetic stand. We then added 80% ethanol to wash away any impurities. We used the indexing PCR to tag each PCR product so that when it is sequenced, we know which sample it came from. The DNA strands denatured at 96 degrees Celsius. The reaction cooled at 55 degrees Celsius so primers could bind to their complementary sequences on the single-stranded template DNA. We raised the reaction temperatures back up to 72 degrees Celsius so Taq polymerase extends the primers, synthesizing new strands of DNA. There was 25 cycles in total. Then we performed the second PCR cleanup. We used the fluorometric assay to precisely determine the concentration of our amplified DNA samples after the PCR cleanup. We calculated the nanomolar concentration to prepare our sample for sequencing. Lastly, the nucleotide sequence of each indexing PCR fragments was determined using the Illumina MISSEQ next generation DNA analyzer. Each single-stranded DNA molecule was amplified, and the instrument reported the sequence of each of the original index PCR products bound to the chip. So in our results of UV spectrophotometry, the data for the samples of 1 and 3 produced the best results, while sample 2 and 4 did not have enough DNA. Sample 2 showed signs of contamination because its 260 to 280 reading was over 2, it was 2.17. The amplicon PCR results as seen in gel electrophoresis did not work as expected because it did not show up at all. Only sample 2 was displayed, and it was only a small, slight line. The samples of DNA that we originally tested were not the samples that we ended up with. Our DNA samples at the end were derived from the stool samples of a husky and a guinea pig. For the husky, the total successful reads were 9,234. We found mostly genus bacteroids and unclassified. The most prevalent species were unclassified and bacteroids stercoris. Yeah. For the guinea pig, the total successful reads were 8,581. The most prevalent the most prevalent genus were unclassified and bacteroids. The most prevalent species were unclassified and bacteroids denticanum. For both samples, the main bacteria that was discovered was the bacteroids, which is a genus of bacteria found in the gut microbiome of some animals from around 10 days after birth. The results that we found did not clearly support our question or hypothesis because we did not attain the amount of data that we would need to draw a conclusive answer to our question.